In this web lecture, we're going to wrap up our discussion of evolution at multiple loci by examining one of the most important applications of the concept of linkage disequilibrium, and that is QTL analysis, or Quantitative Trait Locus Analysis. And we're going to illustrate this method using a case study from some research that was done on two species of monkey flowers in the genus Mimulus. So the genus Mimulus includes flowers with a very wide range of floral types that generally come into a couple of categories. Some of them, like this M. louisii that you see with the pink flower, are classic flower types that are adapted to attract bees as pollinators. Bees see much better in the purple, pinkish part of the light spectrum. They don't see the color red very well, so these flowers tend to be purple or pink. And bees also need a place to land when they're going and gathering nectar, so they tend to have large petals that serve as landing pads close to the nectaries, and there's usually just a very broad, short passageway to get into the flower to get at the nectar. So this is a classic bee-pollinated flower phenotype. The other type of flower that you see in this genus is a classic morphology that's associated with hummingbird pollinators. So hummingbirds see much better in the red part of the spectrum. They're attracted to things that are red. So these flowers tend to be red. They tend to have very long, narrow entrances to the nectaries that are perfect for hummingbird tongues. They lack these landing pads because hummingbirds hover. They don't need to land to be able to collect the nectar. So we've got two very classic flower types. But before we go into the details of this research, let's think a little bit about QTL analysis itself and what are quantitative trait loci. We've talked a lot about quantitative traits. Remember, these are traits that are influenced by a number of genes at different loci, and the more genes that are involved, the more you tend to see this normal distribution of phenotypes, a continuous variation in phenotype. And each gene that contributes to one of these quantitative traits is called a quantitative trait locus, or a QTL. So it's one of those genes that has some influence along with a bunch of other genes on a quantitative trait. So one of the things we might want to know about one of these quantitative traits is how many of these loci are involved in producing this trait, how many have an influence, and where in the genome are they located. Both of these questions we can answer by using this QTL mapping technique. But more importantly, we want to know something about what these genes actually do. So what are their protein products, and how do those protein products affect the phenotype? What kind of influence does each protein product have? In order to get at that question, we need to use a separate method called a candidate locus method, where once you find the neighborhood in the genome where the QTL exists by using your QTL mapping, then you can focus in on particular loci in that area and sequence the gene, produce the protein, and be able to study that. So the QTL mapping is basically just going to answer these questions about how many and where. And there are many, many practical applications for these two techniques combined, one of which is genetic engineering, particularly of the food supply. So if we want to know something about the traits we're interested in for crop plants, for example, we want to be able to identify how many of these genes, where are they, what do they do, and how can we fiddle with them to make them produce the traits that we like even more. How can we crossbreed to maximize these traits? Another one is prediction of disease risk. So many diseases are affected by multiple genes, and we really would like to know how many of those genes, where they are, what they do, to be able to figure out how much risk a person has for having one of these disorders. But the application that these researchers looking at monkey flowers had was more evolutionary. Let's take a look at the phylogeny for this genus to understand what this question is. So our two species here, M. cardinalis and M. louisii, you can see by the phylogeny are sister taxa. And so one thing we would want to know is, what was the phenotype of this common ancestor of the two? Looking at this phylogeny of the whole clade, we can see that it was probably a bee pollinator phenotype. Why? Because the next outgroup out is a bee pollinator flower type, and also the most basal group in the entire lineage is also a bee pollinator type flower. So we see one clade that's entirely hummingbird pollinated flowers, and then this one isolated lineage that is also a hummingbird phenotype. So the most parsimonious solution is that the hummingbird type phenotype evolved twice independently, one giving rise to this whole clade and one giving rise to this one single species that arose from a common ancestor that was of this bee pollinated flower type. So what the researchers wanted to know was, what were the genetic changes that occurred that caused this diversification? And specifically, they're interested in an issue that I asked you to consider when you read the chapter on adaptation, this idea of mutations of large effect versus many mutations with small effect accumulating to produce diversification between species. So they wanted to know, was this diversification the result of a small number of large changes or a very large number of small changes to produce this? So this is a big evolutionary question. I also asked you in that prep assignment on the adaptation chapter to think about this idea of evolution working as a tinker, making lots of little small changes to something that is already in existence, versus evolution working as an inventor, creating something entirely new all at once. So this is what they wanted to get at. So how can we use QTL analysis to answer that question? Well, when you find the quantitative traits that cause these differences between the flowers, you can look to see if it is a small number of QTLs, and then using the candidate locus techniques, you can find out if those are proteins that have a large effect on the phenotype or small effect. You can also tell if you have a large number of QTLs that contribute to those differences, and each of them have a small effect. So that is the question that they were trying to answer with this research. So let's take a look at how this is done. First, let's take a look at the general approach of QTL mapping. The first thing you need to do is to find what we call marker loci. So these are going to be scattered randomly in the genome. And for a good marker locus, you need to be able to identify what the alleles are, and you need to be able to figure out what the genotype is for all the individuals that you're looking at. So markers are usually allozyme loci that can be easily genotyped using simple molecular techniques. So these are going to be the knowns, the loci where you know what the genotype is in each individual, you know where those marker loci are located. So this is one of the knowns in this research. You need enough of these markers so that you can be reasonably sure that one of them is going to be close to one of your QTLs. So you want to make sure that each QTL has a marker nearby that's going to identify it. So at the very minimum, you need to have one on every chromosome. Best case scenario is that you have markers pretty densely throughout the genome to be able to clearly identify as many of these QTLs as you possibly can. If a marker is in linkage disequilibrium with a QTL, then you know that one marker allele will be found with a particular QTL allele more often than expected by chance. That's the definition of linkage disequilibrium, right? So remember that it's a QTL that actually influences the phenotype. So the QTL is the unknown. You don't know where it is. You don't know what its genotype is. You can measure the phenotype, and you can measure the genotype of the marker. So it's the QTLs that influence the phenotype, but the QTLs are unknown. The markers have no effect on the phenotype. They're just randomly chosen loci where you can easily figure out what the alleles are. But the genotype is known, and the location is known. So you can use these two knowns. You can measure the phenotype, you can figure out the genotype at the marker locus, and if there's a statistical correlation between the phenotype, which remember is caused by the QTL, and the genotype at the marker, The only explanation is that it's in linkage disequilibrium with the QTL, and that means that it's likely to be close to it on the chromosome. Make sure you understand why this is true. If you don't, make sure you go back and review that part of where we discussed in class why the position on the chromosome is important for linkage disequilibrium. So you can think of these markers as being kind of like the gas gauge in your car. When you run out of gas, your car doesn't stop moving because your gas gauge went to zero, right? The gas gauge is just an indicator of what's going on in the gas tank. Your car stopped moving because there's no gas in the tank. So you can think of the marker genotype as being the gas gauge. It's got to be a reliable indicator of what's going on in there, otherwise it's completely useless to you. The actual level of gas in the tank you can think of as being like the QTL. That's what's actually causing the effect. The phenotype in this case would be whether the car is moving or not. It's the actual QTL that's causing the phenotype. It's the actual level of gas in the car that's responsible for whether the car is moving or not. So let's take a look at how they did this. So in terms of the QTLs that cause this difference in floral phenotype between the bee pollinator phenotype and the hummingbird pollinator phenotype, at all the QTLs that are responsible for those traits, each species is going to be homozygous for whatever the allele is that causes the trait to be like the bee pollinated flower or like the hummingbird pollinated flower. So you've got two parental types that are all homozygous. So these two flowers are never found hybridizing in the wild. They almost never ever produce offspring together. They never cross. But they can be very easily crossed experimentally. So by just hand pollinating them, you can easily cross these two and they have fertile offspring. And so you can take all of these flowers that are homozygous at all of the QTLs and breed them together. They also used marker alleles that were entirely homozygous for one allele in one of these types and homozygous for a different allele in the cardinalis flowers. So we have not only QTLs that are all homozygous, but we also have marker alleles that are all homozygous and homozygous for different alleles in the two species. So we take all these parental homozygotes and breed them to get an F1 generation that's composed entirely of heterozygotes at all the QTLs and also all of the marker loci. We can then crossbreed these F1 heterozygotes to produce an F2 generation, and the F2 generation can be homozygous for either allele or heterozygous at each QTL, at each marker locus, and this is where you get that continuous distribution of traits because you've got this scrambling of heterozygotes and homozygotes at different loci producing different parts of that floral phenotype. This is the generation on which you're going to actually measure these phenotypes and look for the statistical correlation between the genotype at the marker and the phenotype of the flower for different floral characteristics. Let's look a little more in depth at how this works.